Section for our chemistry of life unit, we're going to focus on proteins and nucleic acids, so the last of the two macromolecules. So proteins, these are polymers with important structural and metabolic roles. There's so many proteins in the human body, and we're going to learn a lot of these and talk about a lot of these, and you'll learn the different roles in a little bit more detail in this class than you did when you were a freshman in freshman biology. So here are some major functions of proteins. They can be enzymes. Those are catalytic molecules. We'll designate a lot more instruction time to enzymes specifically because they're so important, but we'll talk about that some more when we get into the big energy processes for photosynthesis and respiration. Next ones up are defensive proteins such as antibodies, hormonal and regulatory proteins. Those control physiological processes. We'll talk about those in cell signaling. Receptor proteins, again, we'll talk about that in cell signaling. Those receive and respond to molecular signals. Storage proteins, those can store amino acids. Structural proteins, physical stability and movement, that's what that's helpful for. Transport proteins can carry substances such as hemoglobin, can carry oxygen. And then genetic regulatory proteins can regulate when, how, and to what extent a gene is expressed. So a lot of these enzymes, they're going to get their heyday in their own units. So we'll get back to that. A lot of these proteins, my bad, I think I said enzymes. A lot of these proteins, we'll talk about those in detail later. So what's the monomer of a protein? The monomers of proteins are amino acids. So I like to call them AAs, or I like to write down AAs because I'm a little lazy and I don't want to write down amino acids every single time. So protein monomers are amino acids. We have, and you look at this picture here, get a different picture, remember a different color. So we have this picture here of an amino acid. You'll see that it has an amine, an amino group, and it has a carboxyl group. So two functional groups, right, that you're supposed to know and love. So the amino and carboxyl functional groups allow them to act as both an acid and a base, allow these amino groups, amino acids. So that's kind of cool. So amino acid R groups. Remember R groups, the other stuff. So the R group is the side chain. That's going to differ in each amino acid. There are only 20 amino acids that occur extensively in the proteins of all organisms, and they are grouped according to the properties conferred by the R groups. So we have the 20 amino acids in proteins, amino acids with electrically charged hydrophilic side chains. Those are going to be arginine, histidine, and lysine. And then we have some negative ones, aspartic acid, and then glutamic acid. So these ones that we care about, what I'm talking about, these different side chains, those are going to be the ones that are boxed in purple. So those are the side chains in these amino acid groups. So these are kind of similar to functional groups in that they confer a different property to that amino acid whenever they're attached to it. So some more amino acids and proteins. We have amino acids that are polar but uncharged side chains, so they can be hydrophilic. So we got serine, threonine, asparagine, glutamine, and tyrosine. We got some special cases such as cysteine, glycine, and proline. And then we have amino acids with non-polar hydrophobic side chains. So alanine, isoleucine, leucine, methionine, phenylalanine, tryptophan, and valine. Okay. Do I expect you to memorize all of these? No, but just know that these are groups can confer special properties to these amino acids, and if they have unique properties, that can allow them to interact differently with each other or with other functional groups or molecules. So the specific roles of amino acids in peptide chains. So the glycine side chain is a single hydrogen atom, which means that it's small enough to fit into tight corners in the interior of a protein molecule, because proteins are really highly folded, and we'll talk about that some more in a second. Proline has a ring structure that limits its hydrogen bonding ability and its ability to rotate. It often functions to stabilize bins or loops in proteins. So that's kind of special about proline. So proline is special, and glycine is special. And then cysteine. Cysteines are special because those side chains can form covalent bonds known as disulfide bridges, which are important for protein structure. We'll talk about that in a second. So we have this going on here, this disulfide bridge, two sulfur atoms, to build that disulfide bridge, which we'll talk about that in 1 second. So we have the monomers of proteins are amino acids. Well, now we have some other terms. We have oligopeptides, which you should be thinking oligosaccharides. It's the same idea, but with amino acids. And then we have polypeptides, which is the polymer. Again, very similar to polysaccharides, but polypeptides are unique to proteins, this term. So oligopeptides, or peptides, are short polymers of 20 or fewer amino acids. There are some hormones and signaling molecules that are oligopeptides, and those are kind of important ones too. Polypeptides are larger polymers. The functional proteins are made up of one or more polypeptides. So proteins range in size from insulin, which is about 51 amino acids, to huge molecules such as the muscle protein called titin, with 34,350 amino acids. So they got a big range here of how many amino acids they actually have. So I have an example of an oligopeptide. I have insulin right here, not that many, but you can see that these amino acids are interacting with each other as they're folded. And then we have some oligopeptides kind of showing you the importance of these oligopeptides in cell signaling, which again we'll get to when we have the cell signaling discussions. So you're seeing this word peptide, right? Especially in polypeptide. Peptides is a type of bond. So we have peptide bonds. That's what links amino acids together in those condensation reactions or dehydration synthesis reactions to form those peptide bonds. So polymerization, the creating of this polymer, takes place in the amino to carboxyl direction. So we have this picture here with this caption. In living things, the reaction leading to a peptide bond has many intermediate steps, but the reactants and the products are the same as those shown in this simplified diagram. So we have one amino acid in this tan box, and it's being added to another amino acid in this tan box. So you can see that the carboxyl group of one is interacting with the amino group of the other. And what's in the way here? We've got some friends in the way. We've got two hydrogen atoms in the way. Those got to go. And we have an oxygen atom in the way that has to go as well. And when they come out, they form that H2O, that water molecule. So once those things pop out, those atoms get out of the way, then we can form that peptide bond between the amino acids. And then we can continue building that up until we get a little oligosaccharide, and keep building that up some more until we get a polysaccharide. My bad. Oligopeptide until we start building that up and we get a polypeptide. Goodness. Thinking about carbs and proteins at the same time. So we have 4 structures of proteins, and these structures, you do need to know these structures, but kind of focus on what makes them different. Why is a primary structure different from a secondary structure, and what's important in that structure, okay? So the first structure is known as a primary structure. That's basically just the sequence of amino acids. What are its amino acids? And then the bonds holding them together are going to be those peptide bonds, right? Peptide bonds. So basically the sequence of amino acids is the primary structure, just the sequence of amino acids, not any folding, none of that, right? Secondary structure is the regular repeated spatial patterns in different regions resulting from hydrogen bonding. So we got some different ones here. It shows you here, and this picture is a little blurry, and it's pulled from your textbook, so I would just go to the protein section of your textbook to see this picture that's a little bit more clear, because I know this is blurry on here. But in this picture, we have this side, we have the alpha helix side, and then we have the beta pleated sheet side over here. Right, so you can see the alpha helix. What you got going on here is a spiral situation, kind of like DNA. We have the helix, right? It's a single helix. I don't think DNA, so it's double. And then we have this beta sheet, and the way that it is on top of each other, just like a sheet, right? So the beta pleated sheet. So in this alpha helix, we have a right-handed coil, and we'll talk about this right-handed, left-handed business later. And then beta pleated sheet, where 2 or more sequences are extended and aligned, all right? And so those are going to be the secondary structure. These are the regular repeated spatial patterns, right? Either that pattern is a helix or that pattern is a pleated sheet. Pleated, you could think of folds like a pleated pants that got a fold in the pants, okay? So that's primary and secondary. The third one is tertiary structure. This is when a polypeptide chain is bent and folded, results in the definitive 3D shape. So now we're getting into this 3D business, right? And in this 3D business, you can see here, you can have regions of this tertiary structure that has the alpha helix, and then you can also see regions of it here that has the beta pleated sheets within the same protein that we're looking at, right? So that tertiary structure can combine secondary structures and beta pleated sheets to create this new and more complex looking protein, right? But these are the same protein, right? So the same polypeptide has different shapes and it's all interacting with each other. Versus quaternary structure, that's when you have two or more polypeptide chains that bind together by hydrophobic and ionic interactions and hydrogen bonds. So if we look at this quaternary structure, and again, you can see this better in your textbook, you can see that there are different subunits. So we have maybe this gray polypeptide, and then we have this red polypeptide, and this brown one, and then this yellow one. So those are different polypeptide chains, each with their own tertiary structures within it, right? Interacting with each other to build this protein complex, right? And that's going to be the quaternary structure. So these are hierarchical, these structures. They all obviously have a primary structure. They need to have those amino acids bound together. That's the primary. And then whenever those start getting in a pattern, then that's going to be secondary. Whenever you're looking at a protein that has both types of patterns or is just complex, then that's the tertiary one. And then when you're at the quaternary structure, you have multiple polypeptides interacting with each other with their own tertiary structures, with their own secondary structures, and with their own primary structures, right? So it gets more, we've got levels of complexity here for these proteins, one being the least complex to the fourth level being the most complex. So in quaternary, we're saying that there are interactions going on there, and in tertiary and secondary, we got some interactions going on. So where are these interactions actually happening? These interactions are happening between the R groups. So interactions between the R groups determine tertiary structure, okay? So interactions between the R groups that we care about determine this tertiary structure. You might want to try to associate that with tertiary, okay? Disulfide bridges hold folded polypeptides together. Hydrogen bonds stabilize folds. Hydrophobic side chains can aggregate in the protein interior, so they can be inside. And then van der Waals interactions are going to happen between hydrophobic side chains. So this is all happening in the tertiary business, right? And then if it's happening in the tertiary and we have a quaternary structure, then that's happening there too, okay? So what's really important about proteins too is this business known as denaturing or denaturation, right? So denaturing is when heat or chemicals disrupt weaker interactions in a protein, destroying secondary and tertiary structure. This is going to leave primary structures intact, right? So the protein can return to normal when cooled or the chemicals are removed. All the information needed to specify the unique shape is contained in the primary structure, so it'll go back to its secondary or tertiary structure. Unless it's a lot of heat, like a crazy amount of heat is applied to it, or some really, really strong chemicals are applied to it, then it could break down the primary structure. And when the primary structure gets broken down, that's known as degradation. And that is different. That is not the same as denaturing. All right, so degradation is destroying the primary structure. Denaturing is altering the secondary and tertiary structure, preventing that from happening. But as soon as you get rid of those chemicals or heat, then those secondary and tertiary structures will come back. Unlike in degradation, you mess up the primary structure, there's no coming back from that. So what can lead to denaturing or denaturation? Factors that can disrupt the interactions that determine protein structure denaturing is temperature, change in concentration of hydrogen ions, so pH business, high concentration of polar substances, or non-polar substances. If you look at this picture here, you can see what we have, what a denatured state looks like versus its native state. So we have an active site right here, and something can fit in there, right? Maybe we're thinking of an enzyme right here. So I have its active site, it gets denatured because of one of these variables, and now this protein is all wonky and crazy looking. Its structure has changed, and now this active site is gone. It's not where it's supposed to be anymore, and whatever job this protein is supposed to do can't do its job anymore because it is all crazy looking. So back to some more protein interactions, because this is going to get really important for enzyme stuff later on. Proteins can interact with other molecules, such as enzymes. R groups on the surface may form weak interactions, such as hydrogen bonds, with groups on the surface of another molecule. This can change the tertiary structure and thus the shape of the protein. Protein structure can also be modified by covalent bonding of a chemical group to a side chain of one or more of its amino acids. So proteins are very malleable. They can change depending on what they're forming interactions with because of those different properties of functional groups and the different properties of the R groups. All right, DNA. And nucleic acids. So nucleic acids are informational macromolecules. Nucleic acids are polymers that store, transmit, and express hereditary information. DNA is deoxyribonucleic acid, RNA is ribonucleic acid, and the monomers in nucleic acids are nucleotides. Nucleic acids, DNA, RNA would be the polymer. Nucleotides versus nucleosides with an S. Nucleotide is a pentose sugar plus a nitrogen-containing base plus a phosphate group, whereas nucleosides with an S is a pentose sugar plus a nitrogen-containing base and no phosphate group. Right, so there is a difference between them. So the nucleotide components, we got different bases. So the bases are either going to be a pyrimidine, which is a single ring. So we can see here, cysteine or cytosine is a pyrimidine, thymine is a pyrimidine, and uracil is a pyrimidine. They have one ring. You see it, right? And they have sugar. So DNA contains deoxyribose, which is here. It's a pentose, remember? And RNA contains ribose, which is here, which is another pentose, right? 5-carbon sugar. So what are the bonds in nucleotides? Nucleotides bond in condensation reactions, dehydration synthesis, to form phosphodiester bonds, right? So you should associate phosphodiester bonds with nucleic acids, right? A phosphodiester bond is a linkage between the 5-carbon of one ribose and the 3-carbon of the next ribose. So if you're looking at here, we could see this number 5 and this number 3, right? The 3 position and the 5 position. So whenever they form a bond between that 5 and that 3 position, then that is known as the phosphodiester bond. And nucleic acids go in a 5' to 3' direction. When we get into DNA synthesis and all that fun stuff, we'll get into the specifics of that later on and what that means. Okay, but I mean, you can see it here. It goes from a 5 to 3. We love these oligos, right? So now we have oligonucleotides. We had oligosaccharides, oligopeptides, and now oligonucleotides. Oligonucleotides have up to 20 monomers. We have an example of these are small RNA molecules, which you'll learn are important for DNA replication and gene expression. So we'll get back to those later on. DNA and RNA are polynucleotides and the longest polymers in the living world. Okay, fun fact. So how do these bases pair with each other? Base pairs are linked by hydrogen bonds. Okay, we've got hydrogen bonds going on here. So we have a T double bonded to an A with hydrogen bonds. And then we have a cytosine triple bonded to a guanine. So C triple bonded to a G. Okay, so these base pairs are linked by hydrogen bonds, and these are favored by the arrangement of polar bonds in the bases. There are so many hydrogen bonds in DNA and RNA that they form a fairly strong attraction, but not as strong as covalent bonds, right? Thus, base pairs can be separated with only a small amount of energy. We can break those hydrogen bonds, and our cells are always doing that so that they can unzip the DNA and carry out DNA synthesis. The base pairing rules for DNA, and we'll get into this again when we get into DNA synthesis in detail, is we have purines bonded with a pyrimidine and purines bonded with a pyrimidine and A to T, right? So purine, pyrimidine, purine, pyrimidine. So G to C, A to T. RNA is cool. Its base pairs are A and U and C and G. RNA is usually single-stranded but may be folded into 3D structures by hydrogen bonding. And then folding occurs by complementary base pairing, so the structure is determined by the order of bases. So you can see this here, that this is all one piece of RNA, but they're folded about itself, right? So these folded areas, in these folded areas, the RNA is binding with each other, complementary. DNA is usually double-stranded. 2 polynucleotide strands form the ladder that twists into a double helix. And then the sugar phosphate groups form the sides of the ladder, and the hydrogen bond bases form the rungs, if you're thinking of the ladder. So we have the sugar phosphate, a pentose, and a phosphate. That's going to be the backbone, the sugar phosphate backbone. And then in the middle, we have the nitrogenous bases, and those are the rungs, if you're thinking of a ladder. Okay, so if we're thinking of a ladder, we've got the rungs going across. Those are the nitrogenous bases. So DNA is antiparallel. So the 2 strands are antiparallel, which means they're running in opposite direction. And the double helix is right-handed. If you wanted to know how it's spun, you can use your hand to figure that out. So DNA has 2 functions, 2 big functions. Its information is encoded in the sequence of bases. And DNA has 2 functions. Its job is to replicate, and its information is copied to RNA and used to specify amino acid sequences in proteins. And we get into protein synthesis, you'll get into all that fun stuff again.